Hey everyone, Azim here. We are taking a look at sarcomere models for skeletal, could also be for cardiac muscle. So whether we look at cardiac muscle, you have sarcomeres here. Sarcomeres are these repeating segments found on myofibrils within the muscle fiber. Same thing here in skeletal muscle. Repeating segments along a myofibril within the whole muscle fiber. And that's what we're seeing here. First, what are these things represented around the sarcomere? In yellow, these are representing the T-tubules. T-tubules are those little tunnels that dive in from the sarcolemma and then go deeper in and surround every single myofibril within the muscle fiber. The purpose of the T-tubule, the function of the T-tubule, is to spread electricity more evenly deeper into each muscle fiber. Here in green, this is representing the sarcoplasmic reticulum. The sarcoplasmic reticulum is that specialized organelle found in muscle fibers, shown here in blue wrapping around the sarcomere, shown here in beige wrapping around the sarcomere. It's the specialized endoplasmic reticulum. Its purpose is to store calcium, and when a muscle fiber is stimulated by a neuron, it releases calcium, and that leads to contraction. So, let's take a look at the sarcomere with these removed, looking deeper to those organelles. I have two sarcomeres here. I'm trying to show them side by side. So this is one sarcomere, and here's another sarcomere. There's actually two different models, but I'm showing them in this angle so that we can see that this is one Z disc. This is called a Z disc, and it helps that it's zigzaggy. Here's a Z disc. Here's another Z disc. Here's another Z disc. Z discs form the borders of sarcomeres. The middle of each sarcomere, that is the M line. So M line, Z disc, M line, Z disc, and so on. We call the M line, it's the middle. M line's the middle. So when a sarcomere contracts, because sarcomeres are the contracting units of every myofibril, things are going to move closer to the M line. These Z lines, or Z discs, same thing, sorry. Z line, Z disc. These Z lines move in closer to the M line during contraction. It gets shorter. When we relax our muscle, it goes back to rest. So one thing that we can say that happens during contraction is that these Z lines move medially towards the M line. The Z lines move medially towards the M line. What else do we have here? We have structures that appear blue and structures that appear more red. The blue structures, these are the thick filaments. The red structures are the thin filaments. Thick filaments are made up of myosin. The protein that's found in the thick filaments is myosin. Myosin is this long protein. There's a bunch of them bundled together here. This long protein with these special heads. It's these heads that are functionally important because it's these heads that will bind to these red spheres here of actin. These myosin heads form cross bridges with actin. So all these little heads, their goal, so to speak, is to grab and pull on those actin heads. Or I shouldn't call them heads, on the actin molecules. The myosin heads grab and pull on the actin, pulling it closer to the M line. Because there's so much more myosin, because it's a thicker filament, it appears darker. So when we look under the microscope, when we see the darker striation, that's this. From end to end of thick filament, that is an A band. So an A band would be from here to here. It would be lighter here, and then from here to here. So A band a lighter band, which is called an I band, which is only thin filament, A band, I band to the next one. Once again, we only have the A band where we have thick filament. So from here to here. I band, the lighter band, is from here to here. A band again here to here, and so on. I band here and on. So I bands is where we only have thin filament, and at the middle of the I band is where you find that Z line, the Z disc. Looking closer at the thin filament, we can see that these little actin molecules, it's called G-actin, these little actin molecules are arranged in this kind of helical formation. They're supported like on a skewer, which is not shown here, but the protein that supports the actin and arranges it, it's called nebulin. Nebulin helps keep it in line. There is a protein that keeps the thick filament in line. It attaches from the end of the thick filament to the Z line. It's also not shown here. That's called titin. It's called titin because it's the largest human protein. So titin stabilizes thick filaments, nebulin stabilizes thin filaments. What you can see here, besides the red actin, is this yellow molecule and this green molecule. The yellow molecule is called troponin. Troponin is a regulatory protein. I'm sorry, yellow, let me say that one more time. Yellow is tropomyosin. There we go. Yellow is tropomyosin. It's a regulatory protein because what it does, it gets in the way between myosin heads and actin. It prevents them from getting too close. Myosin heads are prevented from getting too close to actin. I'm sorry about the focus here. Myosin heads are prevented from getting too close to actin because tropomyosin gets in between them. Troponin is associated with tropomyosin. When you have calcium released from the sarcoplasmic reticulum, calcium is released into the sarcoplasm, binds to troponin. Troponin pulls tropomyosin out of the way. When I say out of the way, that means now the myosin heads are free to grab onto the actin. And we complete what's called a contraction cycle. Myosin heads will grab onto the actin and then pull, and then grab onto the next one and then pull, and then grab onto the next one and then pull. And that's how we get contraction. I'll say that one more time without messing up. Calcium gets released from the sarcoplasmic reticulum, binds to troponin. Troponin pulls on tropomyosin, moves it out of the way so that these myosin heads can bind to actin. When myosin heads bind to actin, they complete a power stroke, part of the contraction cycle. They grab and pull, grab and pull, grab and pull, depending on how much calcium has been released. And that's what causes contraction. If we want our muscle to relax, then we need to get rid of calcium. Calcium needs to unbind from troponin. We need to put it back in the sarcoplasmic reticulum, and that requires pumps, something you'll discuss more in physiology. We mentioned how the Z lines move medially to the M line during contraction. The A band, since this is part of our center, nothing happens to the A band. The A band stays constant during contraction. The I band, where we only have thin filament, since the thin filament is overlapping more with the thick filament, the I band does get smaller. The brighter band does get smaller. The thick filament, we can get even more specific from what we see here. For the thick filament, we have this zone of overlap. This is called the zone of overlap. Even at rest, there is at least a little bit of overlap between the thin filaments and thick filaments. The zone of overlap will get bigger during contraction. And there's two zones of overlap. Zone of overlap on this side, and a zone of overlap on this side. The zones of overlap get bigger during contraction. This middle part, where you only have thick filament, it's still dark under a light microscope, but if you have an electron microscope, you can see it with a lot more detail. It appears brighter. I mean, it's darker than the I-band, but it appears brighter in this middle part surrounding the M-line. The word for bright in German is heller. This is the H zone. And so the H zone will get smaller during contraction. So to recap all the things that happen during contraction regarding all these funny letters and names, these Z lines move medially towards the M line. They move in. This I band will shrink. I band gets smaller. A band stays the same. But within the A band, we have the zone of overlap. That gets bigger, and the H zone gets smaller.